Okay, so I'm gonna go ahead and apologize, I think, because for some of you, maybe I'm gonna go into like break it down. Hopefully, for somebody who is like kind of knowledgeable, maybe totally in and over their heads. So for some of you, this might be a review, but let's just do it. Let's see, let's pull up the screen, application window. Yeah, what is up? Okay. Can everybody see this PowerPoint I've pulled up?
It's visible for me.
Sick. Awesome. All right, cool. So we are talking about the electrical sensing of phosphonates. What does this mean? Basically, they're going to be using what? Full screen. Full screen. Okay. All right. Whatever. No. I don't know how to use PowerPoint. Obviously. There we go. Cool. All right. So the basic idea here is that the authors of this paper have made a fantastically powerful sensor with some relatively cheap materials. You know, so overarching, I think it's always just best to kind of go through the entire procedure and then get into the details. So, broad strokes here, what they did was they grew up a bunch of their protein of interest, PHND, which is a surface binding protein. So they cloned it, they made some changes to it, they grew it, they grew a whole bunch of it. They made their scaffold that they attached their PHND to. They conjugated their PHND to the scaffold, which is just a fancy science word for they had their protein, they had their scaffold, they made the Lego pieces to smack one onto the other. And then same thing for coupling of proteins to the scaffold. Um, Same thing again. They just smacked more Lego pieces onto there, and then they tested its capacity to do electrical sensing. So what does any of that mean? Who knows? Let's get into it. So the idea is pretty much that biomolecules act as really good switches. And we're going to go specifically with the metaphor of Pac Man here. So think of our protein pH and D as Pac Man, right? He is just a protein that has been isolated from a prokaryotic cell. So, and he's really just he's good at binding and unbinding. That's all he does. He has one binding site. He just wants to eat his cherries and go about his business. So we can sense that. Like we can use that to our advantage and use that as a sensor. So, Pac-Man, his mouth is open or his mouth is closed. So that is our on-off switch here. And based on what we know about how he functions normally in a cell, we can use him as a sensor. So, if we change the ligand concentration, ligand is just, fancy science word for cherries that Pac-Man likes to eat in this case. So, um, so if we change the amount of ligand, so if Pac-Man has just like a ton of cherries in the room with him, that's gonna like change. We're going to be able to tell the difference between a room full of Pac-Mans and like one cherry, right? If we change the pH, that's going to alter the way that they bind, if we bind them to other surfaces, et cetera, et cetera, et cetera. So based on how these sensors are made, you can measure the enzyme activity, the affinity of the ligand for your biomolecule charge, all that jazz. But basically, so you can isolate a piece of a protein and use it as a switch, or you can basically take a chunk of a protein and you can modify it to act as the sort of switch that you want to see, because obviously we're doing weird stuff that cells are not normally doing. But that's the basic idea there. So what is this PHND protein? Or actually, do we have questions? How's this falling on everybody? You good? Okay. All right. So, cool. We have a Pac-Man. Neat. What does that mean? Well, our Pac-Man is a protein. So to jump into that, so this is just an example. This is not our protein right now, but so this is a monomer. This is a single chain. And proteins are composed of domains. So a domain is a sequence of amino acids, about 50 to 250 amino acids long, and it is independently stable. So if you were to
Can anyone else hear her?
Disappeared here as well.
Okay,
She got
just
up.
me.
Yeah, I don't know where she went.
the structural integrity of the rest of the protein,
You,
or we could just
you
cut them off, and that would be fine too.
you cut out for a large chunk there.
Oh no! Can you guys hear me?
Oh. Has anyone else lost the video?
Yeah, that was us. Our internet fucked off. Sorry.
Can you guys hear me?
Yep,
Yeah.
we can hear you now.
Okay.
You're no
Great.
longer streaming.
You need to restart the live stream.
Yes. I understand. Okay. What part did I lose you guys?
Probably asking for questions.
Probably asking for questions. Did everybody see this slide? Did anybody have questions at this point, or should I go back and explain?
I think we made it to the monomer slide just fine.
On our slide. Okay, great. Okay, so we have our monomer. It is composed of domains. Each of these domains is stable on its own. So we could chop it off from the main protein, or we could modify it, and everything would be fine. So that's the basis here. So our PH&D protein specifically is composed of two domains, and it's kind of like a clamshell. So we can really mostly think of this as Pac-Man still. The metaphor holds as a switch. You know, Pac-Man is eating cherries. Pac-Man does not have cherries, but also it does kind of actually physically look like that, which is cool. So the idea here is that Pac-Man has two conformations. So Pac-Man's mouth is open, and the hinge regions are 70 degrees extended apart from each other. And then when Pac-Man encounters a ligand, or cherries, one of those regions rotates by 70 degrees up and closes. And so when this occurs, the protein itself experiences a 7.7% reduction in surface area. And so that is really important. Keep it in mind. We're going to come back to that. Yeah. So let's talk about ligands. Let's talk about Pac-Man's cherries. So in nature, phosphates are just freaking everywhere. They're absolutely everywhere. So 2-AEP was actually the first phosphate to be discovered. So it's this structure in the middle here. It is present everywhere in the soil. Bacteria basically use it to break down available phosphorus, nitrogen, oxygen, all that jazz for use. But phosphates in general are also just everywhere in the biomedical industry. They're used as additives in manufacturing. They're just everywhere. But so this structure here is also called a bioisomer, and the idea is basically just that it is a structure that has something like phosphorus on it that can be broken down for later use, because obviously elemental phosphorus or elemental nitrogen or oxygen are super reactive, and you would never be able to deliver an element like that to the place that you needed. So it's just, it's kind of the packaging that biology uses to deliver some of these elements from one place to another. So phosphates are characterized by, we've got a phosphorus group in the center. We've got two OR groups. We've got a random R group, and then we have are double bonded oxygen. So all phosphonates share this structure. 2-AUP, first discovered phosphonate. From there, we just kind of unraveled everything else. And in my research in my lab, unrelated to this paper, we were modifying the same protein, PH&D, in order to use glyphosate as a ligand. And as you can see, from left to right, the complexity and the size of our structure is increasing. So, of course, your ability to make modifications gets a little bit more complex as well. Any questions?
Yeah, I got a question, but it has to go back with the Mortimer?
Yes.
So I'm a little confused. If you can bring that slide up again, I guess. Hold on, maybe I can go back to it. There we go. Okay, there. So you're saying that when the okay, my brain's trying. So the green part is the Pac-Man, and the blue part with the little red inside is the cherry. Is that what you're saying? And then, is that, first of all, is that right?
Super, super close. Okay, so on the right-hand side, this green part is the PH&D, and on the right-hand side, this blue part is also the PH&D, and then the red part is the cherry. So what's happened here is that the protein itself has folded in to encompass the cherry, and now it's holding on really, really tight. So the cherry is in Pac-Man's mouth. Pac-Man has closed his mouth. That lower domain has rotated 70 degrees to fold over the cherry.
Okay, so it changes color. It has decided, yum, this is terrific. And then it just needs to turn over as well because it just ate a cherry. It's very exciting.
The color is really just for us to say, this structure has changed somehow.
Okay.
But it's the same.
It does move around.
Yes. Yeah. And proteins move around a lot. This one only moves in one type of way. He opens his mouth or he closes his mouth. There are other proteins that move in a lot of different ways. But this is really
only does it 70 degrees.
Somebody figured that out through X-ray crystallography, and
I don't even
that
know
is a whole other.
That is such a cool word.
It's super, super cool. It is not this paper, but somebody did some really neat science to figure that out.
Okay. All right.
Yeah.
Thank you. Okay. Thank you for explaining that.
Yeah, for sure. Okay. And this structure, generally speaking, is called ribbon structure. And it's the way that we visualize proteins of this size because proteins have primary, secondary, tertiary, and quaternary structures. And so what we're seeing here is all of those structures in this emergent shape, basically. And it's a whole lot of biochemistry. It's a whole lot of molecular bio. I'm sure we will make some resources and have some like more in-depth chats about why this looks the way that it does. But it's really, really cool. We'll get there, just not today. All right, moving on. So phosphonates. This is our cherry. This is what our protein likes to bind to. Everybody's good with this concept?
Yes.
Sweet. All right. So what they did, they made a solid state nanopore. So in this case, solid state doesn't really mean what it means for other technologies. Basically, what it just means is that they made a foil that was not biologically derived. It is just plastic and metal. That's it. So the way they did that is they had a polyethylene something film. There's the name. The name's not really important, but they had this film, and they had this machine where they basically kept it on this side on the right hand here, and they basically just had a giant electron gun that they put gold into. So on the left here, if they put in gold, they accelerated it super, super fast with a magnet. They defocused it a little bit. It came through. This is where our foil would be. They basically just shot it with a teeny tiny electron gun to make holes in it. So on the left hand here you can see the different types of substrates that can be made. So you can control it so that you have just a single pew. You can make like really straight lines, or you can make a pattern. If you keep it going, or if you move your foil in such a way, you can basically shoot it more, and so you have holes that are not overlapping. Or you can just hit it with your shotgun and just like completely obliterate it, and then you have this very how would you say? Anyways, you would just like shoot the crap out of it, and then it would just really be chewed up on the right here. Questions? Cool. All right, so super basic. They bought this foil, they shot it. They're like, this is great. It's got a bunch of holes in it. Fantastic. Moving on. So how did they confirm that they got the pattern that they wanted on the foil? Well, what they did was they seeded it with GFP. GFP is very similar in size to AEP, so and PH&D. So if PH&D is around 37 kilodaltons, GFP, I'm sorry, yeah, PH&D is around 35 kilodaltons. GFP is around 27 kilodaltons. So what they did was they soaked their foil in a solution with GFP. And they're like, some of this GFP is going to make it into the pores. And so then they basically looked at it under conditions that would make the GFP fluoresce. And they're like, oh, this is great. We can see all this fluorescence. Obviously, there's GFP inside of the pores. This means the pores are present. Fantastic. So that was how they confirmed that they had done this step properly, which is great. This paper is really, really fantastic about like checking their work at every step. Questions? All right.
Okay. Seriously, I know that some of you people have no idea what the fuck is going on, so ask questions.
Well, we'll ask questions at the end. It's fine. Okay, so this is a really terrible picture. How did they confirm that their PH&D was working at all? Well, what they did was they seeded their new foil with it, and they ran their experiment to see whether or not the PH&D was binding or not, and then they ran it again after thermally decoupling or denaturing their protein to make sure that it was the protein itself that was doing the binding activity and not something else that they weren't considering at the time. So this is a scary graphic. Don't worry, it's not that scary. So the black line here exhibits a very characteristic binding curve. So when you get into it, biology has a lot of sigmoidal curves, and you find it absolutely everywhere. What sigmoidal is based on a logarithm scale. So biology, you can't make faces like that. That's terrible. Whatever. Anyways, this is a very characteristic binding curve. So seeing this, you're like, excellent, it works. That's great. And then they denatured it. So basically, they put their PH&D in the microwave. They increased the temperature. They broke it. What happens is basically you excite your protein to the point where the chemical bonds that give it its structure and shape are so excited they break, and then your protein itself unravels. And if you don't have that 3D shape anymore, it's not functional. It just doesn't work. So they confirmed that their protein was doing the action that they suspected and not something else. Excellent. Questions?
I know that some of you have questions. Ask your questions.
Can't force them. Okay, so functionalization. So at this point in the paper, if you read it, you might notice that they basically said each of these steps and referenced another paper, and that was it. So this is Journal Club. We're going to cover this paper. They functionalized their foil. This part wasn't necessarily research. If you want to learn about the nitty-gritty, about the chemistry that's going on, you should read those papers for sure. But basically, we can just say, like, they didn't give us the information. They didn't think it was important enough. So we don't really think it's important enough. Like, this is just kind of tried and true stuff. So we're just going to kind of gloss over this one. This is how it happened. There's the papers. The papers themselves are referenced in the paper in the library. So if you are super curious about this, Jazz, go for it. Here's another really terrible graphic. I got some tips on how to take better graphics. But basically, this is just the picture version of the previous slide. So this is all the chemistry that happened. And yeah, so they had their original foil. They modified one of these carboxyl groups to have an amine ending. That was one step. After that, they treated it with the nickel, right? And so they smacked on another functional group again and again and again. So basically, this is the step. This blue line with the orange dots are the coupling. And then, so this orange dot here is what the protein itself, so the protein is the purple sphere, attaches to. So this orange group on the right here, this pyridine nitrogen is what the PHND itself attaches to. So chemistry, visualization, it's not super important though. So this was basically achieved through surface-initiated polymerization, RAP polymerization, and this is just a type of polymerizing reaction. What's interesting here is that this specific type of polymerization reaction will run until you run out of reagent, or you take it out of the solution. Otherwise, it will just go infinitely. But
May I?
go for it.
So this is a type of to Luby still. So this is a type of surface modification, and so when you're looking at the previous slide, what you're looking at is that you made a long chain of a polymerized molecule, right? And then this thing wrapped around these objects, and so you have these little orange dots. And the reason that we have these little orange dots is because signal to noise ratio is a big problem in binding. And so you have this very large object that needs to touch your sensor. And so you need to have a lot of them. Am I correct about this? Because this is definitely something that I've seen when people are talking about signal to noise issues. Like, the thing that you want to see is like very small. The thing that lets you see it is very large. And so you need to have a lot of it. So you have with these dots, this is the signal to noise situation.
If I may just say a little tiny thing, the orange square is the NHS group. It's not the National Health Service. It's yeah, that's it.
Yeah, totally. I don't know if they chose this method specifically to reduce signal noise issues, but probably. Okay, so how did they do this surface modification? This was a polymerization, so surface-initiated polymerization. The way that this works is that they had their foil, it had their initial carboxyl group, and they this type of polymerization, basically each chain is built sequentially, so it builds evenly like this. And one of the advantages of this sort of polymerization is that you have really fine control over the polydispersity, which is just a fancy word for how large are the chains that you are building, and are they similar in size to each other? Like, do we have like, and this is sometimes also called a polymer brush, because it kind of does look like a brush, right? So like, are each of the bristles getting larger in size with each other? Is it even? Is one of them going to get very long and the other ones are going to get kind of short? So they all build in length pretty much equally, which is really, really good. And this is just the chemical that they always use for this sort of polymerization event. It's just always there. And like I said before, this is kind of an interesting polymerization technique in as much as there is no termination step. It will not stop on its own. It will run until it runs out of reagent or you take it out of solution, which is kind of interesting. Most of the time, these sorts of polymerization reactions do have a way that they will terminate on their own. Questions? Okay. All right. Well, and so this is just that, but more sciencey. So polymerization, it's kind of like radioactive decay and stuff like that. You have a free radical that initiates a whole set of other chemical reactions that makes these chains grow. So that is the nitty gritty on how that happened. Questions? Okay. So we have our foil. We have it coupled with our protein of interest. Fantastic. So now we have our solid state nanopore with our proteins. How does this work? What are we doing here? So the idea is that we have our foil, we put it in electrolyte solution, and we run a current through it. And DNA is negatively charged, so it's always going to experience an attraction to a positively charged pole. And so we have, so this is just an example. This is not what happened in the paper, but just to kind of explain this concept. So if we had a nanopore and we had DNA in solution, if we ran the current, it would go from one end to the other. And this passing through the pore would change the, oh gosh. Oh, sorry guys. So this changes the current. And so we can see that drop in current and we can say something passed through here, it was sensed, and that change in current is associated with your concentration of protein or even different types of protein. So on the right here, you can see just like in a linear event, maybe just a whole protein or chunk of DNA passed through. You can tell based on the change in current if the protein itself has been folded, if it's got stuff like knots in it, if it is bound to certain things. So it's a very sensitive tool. You can tell a lot of different information in it. So in our case, instead of having DNA here, we would put in something like our 2-AUP, our Pac-Man cherries. Questions?
Anyone need more explanation on how a nanopore works?
Okay, cool. So they ran 4 experiments. They tried it with nanopores with different diameters. They also tried it with ethyl phosphonate to compare. They used a standardized electrolyte solution. They titrated their ligand into the solution. Titrate, just a fancy word for they added it slowly in increments. And we kind of covered this before. They denatured their protein afterwards to make sure it was the protein itself doing the work and not something unexpected. Cool, neat. So how does this work? So in this graphic we have our little purple crescents here. So these little blue dashes, this is our polymer brush. Our little purple crescents, these are our proteins that we attach to those. So if we just ran the current, we would see a certain charge. And then if we titrated in our 2-AEP, we would start to see that it is binding to our protein on the surface here. And so the PH&D experiences a 7.7% reduction in surface area. And so after binding, we would see an increase in current because more electrolyte would pass through the nanopore. And so we can use the current as a function of binding events. And so you'll be able to tell, having done this experimentally, you could theoretically take a sample and put it in and then be able to figure out the concentration of something like 2-AEP or another phosphonate like glyphosate. So that's pretty cool. Questions? Thoughts? Okay. So this is just kind of a larger graphic of the same thing. So here we have our ribbon diagram of the unbound pH&D, and then our phosphonate, Pacman's cherry, bonks into it. pH&D is like, amazing, I love this cherry, it's great, yomp. And it has that 70% or 70 degree hinge rotation that has our binding. So our current increases. We know that the pH&D are doing binding, which means we know that there's 2-AEP in the system. On the right here, we can see the current increase versus the analyte. So again, we have that nice like binding curve that's very characteristic. So we can definitely tell, binding is happening, our protein is working, there's definitely 2-AEP in the system. So the cool thing about this is that the modifications to the protein itself are pretty conservative. It's not too radical. It's not a huge burden on the protein itself. But most importantly, it doesn't require a label to do sensing. So label just means a fluorescent group. Fluorescent groups are awesome. They are a really effective way to do sensing, but they do have their drawbacks in as much as attaching another group to a protein is going to have its own complications. But additionally, when you're sensing things like this, you have to really learn how to use your machine because you can burn out your fluorescent groups if you have your sensitivity turned up too high, all sorts of shenanigans. So it doesn't require that. That's really nice. The proteins themselves are stable when they're by themselves or when they are attached to this nanopore, which is really, really cool because a lot of things act differently when they're by themselves or when they're in groups. Like this is kind of the basis of, chemical sensing and stuff like that. The limit of detection was a lot lower than ion chromatography or amperometric detection. So they found that it was 10 to the 2 to 10 to the 3 times lower. And those machines are sensitive to 1 micromolar, 10 to the negative 34. So that's fantastic that it actually improved upon that sensitivity. Like I said, the protein itself, it was cloned, it was modified. The modification wasn't that intense. Like we didn't actually structurally change the pH&D, so we could consider that a native protein. And the equipment, everything is pretty easy. And like all things considered, it's pretty easy to grow up a batch of this protein and purify it. Like that's like a relatively cheap reagent in lab world. And what's really, really cool about this is it could potentially sense between different phosphonate groups. So And that's really great. So it's easy to tell two very different things apart. It's way more difficult to be able to differentiate between two very similar groups. And so this has the potential to be able to do that. And so what does that mean? Like this is the same sort of like the chemical being sensed here is in the same class of what they're swiping your hands for if you have been in, I don't know, say an American airport and you get flagged for something, you know, maybe you were just gardening and you just have a lot of trace phosphates, you know, on your hands or whatever. And they're like, I don't know, you might be making bombs. So this might be able to differentiate between, this is the chemical that somebody is using as fertilizer versus, this is the chemical breakdown derivative product of a bioweapon. So that's great because it could reduce false positives a lot, which is neat. Theoretically, you could, question? Okay. Theoretically, you could also make this into a portable real-time machine because at the end of it, you really just need a bucket with electrolyte solution to pop your foil in the middle there and to run it and to be able to connect it, you know, to a computer to do all the smart stuff, you know, doing all that math in the program to be like, oh, this is a folded protein versus, you know, a straight protein. But it's pretty cool, and we're probably going to see some more sensors built like this in the future. Questions, thoughts? How are we feeling about this?
Sorry. How resilient are the nanopores? Like, because I know in the like nanopore sequencing, the pores themselves will like pop, but I mean they've kind of plugged it with a protein. Does this have the same problem? Like can you just keep running this over and over again, or does it wear out?
Theoretically, you should be able to just run it over and over again. So eventually, the proteins themselves will denature on their own. So you might have to reseed it with more PHND, but the base structure itself shouldn't break down. But they didn't really test that, so I don't have empirical data for you on that point. Like the purpose was to see, hey, can we sense phosphates? And they can, and they do. So.
Jasmine, I know that you have a list of questions. Would you like to help?
I do have a lot of questions. They're kind of basic, but yeah. One of the first questions that I had. Sorry about that. I was going to my questions. Okay, so there's one point where it says, therefore, one key element is the production and incorporation of a molecular detector to an electrical sensing platform. And I said, why is this a therefore? What is being assumed here about why having a detector would be helpful? Is that because nature knows how to do it automatically, but we're brute forcing it? So if we had something to monitor it, maybe we wouldn't mess it up so much?
So this
That's a good question.
So this has to do with when they're talking about the modifications of a certain site. It's not about nature knowing a thing. It's just about a thing actually working, which is one of those really weird and interesting concepts where, you know, because we like to anthropomorphize, we like to think about things knowing stuff. And so this works as a really good binding site because it does that job. And that's it. There's no intent existing there. And one of the things that BAST has been working on is actually fiddling with those binding sites And so if you would like to talk about some of the stuff that you've been doing with Chimera, I think that would be helpful to answering this question.
Okay. So I'm going to talk and I'm going to pull some stuff up in the background here, so don't mind me, y'all. So proteins are made of sequences of amino acids, and those amino acids basically interact with each other to give the larger shape of the protein. So if we so the PH&D has one binding site, so it's got one mouth. There are proteins that have multiple mouths, if we're gonna like roll with this metaphor. But PH&D only has one, and that's good because it means it's pretty simple, and it means we can mess with it and know really quickly and know exactly like why something is happening in response versus having too many variables to control for. So one of the
So you would say that there's like one very specific Lego that does the job?
there are Legos inside of Pac-Man's mouth that do the job, and so you can modify Pac-Man's teeth basically to have different binding properties. So something else I was doing in lab was basically mutating this protein to try to alter its binding affinity. So let's see if we can pull this up.
Mutating Pac-Man sounds like it's going to have very bitey teeth.
It could. That's the idea.
What if we want bitey teeth?
Well, that's what I'm hoping for.
So there's this program called UCSF Chimera, and it is a protein visualizer, and we can basically use it to try to make experimental modifications to proteins, DNA, all that jazz. So here is the same protein that we were talking about in the paper. And the idea here, so this in the center here, this structure, this is our 2-AEP. So this is Pac-Man's cherry, and he's just kind of like floating in the middle here of this structure. So one domain has rotated to glom onto Pac-Man. So there are teeth in the upper and lower jaws, right? So for example, let's see. So this is one of the teeth. So the idea behind like modifying Pac-Man's teeth is just like we could take this amino acid that is directly next to the ligand, Pac-Man's cherry 2-AEP, and we could change it to another amino acid that would have a different charge so that the 2-AEP and the amino acid would experience attraction to each other. And so like when the 2-AEP got next to this protein, it would experience like a larger attractive force and it would be drawn into the binding pocket, and then binding would be more likely to occur. The other type of modification that can be done is to basically change out amino acids for smaller amino acids in order to allow Pac-Man to eat larger cherries. Because if he can't basically like close that second domain, then binding just won't occur. So something, well, we could just be like, hey, this is a phenyl tyrosine. What if we just made it a straight up tyrosine or something like that? And we could basically just be like, cool, well, you know, what if we just do this? And so there's just physically more room now. So we could like theoretically make a protein that has this alteration in it that has just a little bit more room for this ligand to hang out in. And that should increase binding. So that's the sort of modifications that are happening in these sorts of things.
How do you know which one to go with? Or do you just try both? Like, do you run tests for both? Or does one, how do you know which one to make?
It's a really good question. You basically, So what I did was I picked out 4 amino acids on the inside of the protein here, and then I made a huge list where I was like, here are all the possible combinations of all of these different modifications, right? So, you want to test whether one modification on its own is going to do good work. Maybe if you do 2 modifications at the same time, they're going to conflict, and it's going to be worse than before you started playing with it at all. So basically, you just make all the possible different combinations, and there is other software that I don't know how to use yet that will basically predict the energetic stability. And so you just like have a big long list. You do it all. You test the stability in a new program, and you pick out 4 to 6 mutations. You physically like send out to have those made, and then you bring them back to the lab, and then you just test it, and then you do the same sort of experiments kind of that are in the lab here. So you would run same experiments with the wild type, the unmodified protein, and you would say, okay, was this affinity higher than the unmodified protein, or was it lower than the unmodified protein? And you just do the work. Yeah, so for 4 amino acids that I was considering mutating, that generated a list of 28 possible unique combinations of different mutations I could do to this protein. So it's a lot.
You do this in the lab, you don't do it through like simulation software?
You use the simulation software until you get to a point where you're like, here are my proposed mutations that are energetically stable. I'm going to physically test 4 of these.
Gotcha. Okay.
You use it as a filtering tool, basically.
Right. Yeah, you gotta narrow it down because there's so many
Yeah.
combinations.
Yeah, some of them you can just tell right off the bat. You're like, oh, this is not going to work. This looks like it's just going to fall apart right off the bat. And then you have some of them where you're like, well, it doesn't immediately look like it's going to explode. So let's test it out. And then you just, run titrations forever, and you test its binding affinity. So mentioned in the paper was the KD. So in the paper itself, the research paper, the KD, which is the dissociation constant, is lower than the wild type, but not significantly so. So even though it was lower than the wild type affinity for just straight up to AEP, the scientists were like, you know what? That's okay. It's close enough, and we want to see if we can make this like poop out some like results anyways. And it was within a window that made sense to try it. But this paper is pretty cool in general because I feel like they did some good work basically confirming at every step that they had made what they were looking for. There wasn't really any fluff that there wasn't any sort of like that ambiguous language where you're like, you guys didn't know what was going on, and so you kind of talked around it. It was pretty straight up. There was one sentence in here, let's see if I can find it, where they're talking about binding being relieved inside of the nanopore itself. And that was kind of like, that made my spidey senses tingle because like relieved is a weird use of the word in this term, and there isn't really a like science term to link it back to. So something was happening inside of the nanopore, and they didn't really understand what it was. So they wrote a fluffy sentence about it, which is good. They could have not mentioned it at all. But that fluffy sentence didn't really make sense to anybody. It didn't make sense to me. It didn't really make sense to my boss either, because I was like, hey, boss, what does this mean? He's like, I don't know. That's weird. But other than that, like the results were pretty clear. And the steps that they took were fairly understandable for me, being in this weird niche position that I'm reading this paper casually. But anyways, all that to say, this was actually a good research paper. Are there more questions?
Yeah, so I guess one of the things is that as I'm looking through this and it says in general, towards the end, I don't know. In general, our idea to functionalize a nanopore surface with proteins can be described in 2 steps, in brief, and then it says a whole bunch of stuff, right? And so you had said very early on about how that there's sort of a way to look at this, that if I see all this stuff and there's more stuff that I want to learn about, but then it literally feels like there's another tab in my brain and then like there's another thing to look for. And so how do you determine what exactly you're supposed to, which rabbit hole you're supposed to go down besides what you're just interested in, like to actually learn? Does that make sense?
Yeah, for sure. Well, so in the references, there are basically all the papers that the authors read or actually referenced to explain how did they get to where they think they are now. So in the paper itself, usually, those are labeled 1, 2, 3, whatever. So if you have a specific question about that, so like, for example, the 4-step functionalization, like there are 4 sentences in there where it's literally one sentence and then a reference to a paper, followed by another sentence referencing another paper, so on and so forth. So like that is one way to do it. You're like, I want to learn specifically about this step. I'm going to find specifically that paper and then read up on it. The other part is I think it just takes time and exposure, because some of it you kind of understand what they're saying based on just general knowledge of the field. And sometimes what they're conveying is like a very specific piece of information in a paper. And so it just takes time to figure out which of those they're talking about, because that's going to change how you're going to like search for that piece of information, right? But
no, that makes sense, because literally I was trying to search for if the kind of foil they were using was like the kind of foil I use when I'm baking, or some sort of special science foil. And I'm like, I don't even know how to search this. This is so beyond me.
yeah,
Polyethylene.
In some ways it's so basic, but I don't even have that foundational to know. And so then the internet gets very big.
totally. Yeah, for sure. Let's see. For this one, I believe it was just like a type of foil that you can buy. So let's see. Circular polyethylene terephthalate PET foils. Yeah, so like this is literally just a type of science foil that you can buy. So let's see. I'm on page 18 at the very top under the section fabrication of solid state nanopore. So polyethylene terephthalate, like they bought this. They bought this from Sigma Aldrich, and then they shot it with a gun, and then they slapped some chemicals on there. Like that's it.
Yeah. Also, polyethylene terephthalate is PET. It's the same stuff as like water bottles.
Exactly. Yeah.
Fucking water bottle.
Yeah. It's hard to know like when it's time to zoom in and like go down the Google rabbit hole, and when something has been said already in the paper, but you're just like kind of tired and cross-eyed from all the details, So my advice would be to take breaks for water and to stretch and walk away, and then like try to convey it to somebody else and be like, does this make sense at all, or am I like losing it? But every sentence should be like, every sentence that's actually referencing something should be followed by the number that is in the references. So like the number should be like, and then we talked about this paper, and that should be your first step.
I mean, the major takeaway could be that you make confirmation change, or like you basically change the functions of a surface binding protein to achieve a certain confirmation, like basically a 1 or a 0. Are you detecting this or not? Like that's what they do. The SBP, they detect stuff and send the signal back to the yeah.
So part of what we're looking at here is some of the stuff that I talked about the first time that we did Journal Club. When I was talking about like, you don't actually need to know the names of things. You can just like put in the word foo or bar or just, like the ever loving fuck. But then sometimes you can just look at this long chemical name and you're like, and then you're like, oh wait, PET, that's the name of water bottle. And so then you can just in your head, take the word water bottle, because that's what they did. They're like, we bought some quality grade water bottle and we used it as our surface. Because that's what they said. That's what the paper says. And so, like, don't be concerned about the jargon. The jargon is just a rabbit hole of trying to learn an entire decade of chemistry background. You don't need that, though. You need to just know that PET is water bottle. And then you can just be like, oh, hey, fuck, it's water bottle. Okay, cool. And then move on. And just be like, okay, so they took a water bottle, they shot it with a bunch of things, a bunch of gold particles, and then they were like, pew. And then we had our surface. And, you know, that actually worked. I mean, that's like a whole chunk of that paper is basically we took a water bottle and shot it with gold.
Yeah. Also,
So-
as you like the more of these sorts of papers that you read, like eventually you'll start recognizing stuff like that. So like PET is used for essentially exactly this thing in a huge number of like different applications. Like if you want thin film with lots of little holes in it, PET is just the way to go because it's easy to poke really tiny holes in. So like it's used as a membrane in various types of batteries, for example, because you can make little teeny tiny holes in it that only let very small things through. So like it's, you know, it's you like the more, like I said, the more times you read papers like this, yeah, you see the patterns. Like as like I was
consistency
also
over time and exposure to materials.
Yeah, I got here a little late, so I didn't hear what the film was made of. But literally, as soon as she said polyethylene terephthalate, I'm like, oh, okay, they use PET. Like just because like that immediately made sense. I'm like, oh, okay, that makes sense. Like, of course they use that. Like, so it's just one of those like through exposure you get used to these sorts of things.
The most important thing, though, like the chemicals themselves, less important. The most important thing I think is just being like, because there's a section in here, like where there's 4 sentences and they reference a paper in every single sentence, and that's super overwhelming because, my God, that's awful. Do I have to read 4 papers to understand 4 sentences? But the important takeaway is just like, okay, like they followed a procedure. The procedure's over here. It accomplished the thing. Okay, moving on. And I think really you can kind of understand what's going on by looking at the graphics themselves. You know, so there are a lot of images in this paper. Let's see. I think I'll share my screen again. That and you know, unless you are in the field and you need to find a very specific chemical, or you need to like actually change something about your procedures, unless you're looking for something as specific as that, you can kind of just like look at the images and then tell the story from there. So, you know, here, like image A here, I included that in the PowerPoint, but it's just like, hey, we have a protein, it binds to a ligand. Awesome. Down here in B, they basically confirmed that they made their protein. So they did, they ran a gel, they did a Western blot, they purified it, and then they did titration curves with 2 AEP and let's see, ethyl phosphate. Basically to compare it to their wild types. So, you know, this is just them checking their work. Here's them talking about the general theory. Let's go to some other graphics here. This is just the whole, how did we functionalize this like surface at all? So like here's the specific chemistry, and like literally this graphic is 4 papers. This graphic is 4 papers. Like literally this step to this one is 1 paper. This step to this one is 1 paper. This step to here is another paper. So as long as you can recognize that and not get overwhelmed, you're good to go. Like you don't really need to understand all of that in order to understand what else is going on, you know? These graphics, they're just checking their work again. So they ran a blot, or I'm sorry, they ran a gel to confirm that their protein actually did couple with what they were modifying the PET with. And so they were like, huh, did it work? Did the things bump in together? I can't really see. So this gray arrow is basically just like, hey, the pH and D bound with the that we're coating the PET in. And at the bottom here, we can see where it's just like completely run through, and that really didn't bind at all. That's fine though. That's expected. That's always going to happen a little bit. And they did a mass spec to confirm that stuff was coupled as well. So you can see there's a big change in these black and red lines. So you don't really have to understand everything that's going on, but they saw a change. They knew it was the change they were looking for. Okay, cool. Let's see. And then same thing here, just like, hey, we confirmed that our nanopore actually has the pore size that we're looking for. We saw the GFP in there. Great. We made sure that it was our pH and D and not something else doing the work. So, really just kind of like skate through the graphics. And if you can understand the graphics, you pretty much understand what the paper is trying to say. Everything else is just details that they should include and are good and are very important. But for like I understand in general what's going on in this paper. If you understand the graphics, you're pretty much good to go. Everything else is just extra at that point.
Yeah, in general, it seems like, and this is going to sound so obvious when I'm saying, but it seems like writing the words for science takes up so much more than actually showing the science. Like, there's a lot of science that needs to be explained that literally the graphics show. I don't know.
Totally.
Yeah, so one of the things, and we talked about this in the first journal, but one of the things that there's just an entire language, and the language is very, very precise. Like, it makes philosophy people like tremble, just because it's so intensely precise, because it has to be exactly the thing, because you can show a good picture. Like, I can draw you a picture of an elephant, but you're not going to actually be able to like recreate an elephant. And so the language is the recipe. I can show you a picture of a cupcake, and having a picture, like, you can see the cupcake, the process of a cupcake being made, and you're like, this is what I'm supposed to do. And that helps you understand the science paper of making a cupcake. But a well-written science paper also has very explicit language to make sure that you make the cupcake right. Because you can make something that is cupcake shaped based off of pictures, but it's not going to actually make a cupcake. So you do actually need the instructions, and the world of biology and chemistry has so many more words and ingredients than cooking does, for instance. Cooking is just a subset of chemistry. And so this means that you eventually need to learn a whole new language and then apply that to how you understand the recipes. And if you're lucky, you find a cookbook that has a good recipe. Whereas, we have learned multiple times that you do not always get a good cookbook.
And the main thing about the specifics of the wording is reproducibility, which is a big thing in science. It's a thing. If you can't reproduce it, it never happened.
What's nice about this paper, though, is that they are pretty forthcoming with how they did it, like explicitly what chemicals they used, what steps they used, what techniques they used. And when they don't get into the nitty-gritty of that, they reference other papers. And so this is actually a good cook, or this is a good recipe paper. Like there's sometimes you read papers and you're like, they are obviously trying to parcel this out into 2 or 3 papers, or they are obviously trying to protect some work that they are trying to patent down the line. And therefore they're kind of like wording their stuff ambiguously or not coming forth with all the details. This paper didn't do that. Like I said, there's like one thing that I kind of like squinted at, and that was the odd phrasing, like the coupling pressure is relieved inside the nanopore, which could mean a couple of different things. It isn't really defined in any field that I'm aware of. So they get like, you know, half squint from me for that line, but as for the rest of it, they check their work at every step. They showed the data for checking their work at every step. And that's obviously very good for if you are trying to do similar work and you're like, how do I design good study? You know, it's like, well, here's a great example of designing good study. But despite the pain of reading science papers, this was actually a good one. Boom, Journal Club. How do people feel? Questions?
I guess if you don't really need to cook the cupcake in the analogy, you don't really you can just look at it and say, oh, that's a cupcake, and you don't really need the recipe, right?
Yeah, for sure. And that's kind of what the graphics should tell you.
On the other hand, like, while you don't need to read every paper in the same way that you shouldn't try to hug every cat, having a better understanding of how to make cupcakes, like maybe you don't want to make a cupcake, but maybe you want to make brownies, or maybe you want to make cookies, or maybe you want to make something else. And so having a general understanding of how systems work allows you to, hopefully, using critical thinking skills, to apply that to other situations where you're like, huh, you're not a cupcake, but you're cupcake adjacent, and this is useful. And that is important. And so this is why we read.
So in that analogy, you know, if you were looking at surface modification and you wanted to attach biological things to your surfaces, in that case, I would read those 4 papers in that step. And I would go through that with a fine-tooth comb because that's going to give you the jumping off point for how is this done.
Also, graph polymerization is just like the shit. It's
Yeah, but if you want to learn about polymerization, if you were like actually trying to learn about like how do I do this specific thing, you know, like you would want to read that section with a fine-tooth comb, and you'd want to read a bunch of papers and then start thinking to yourself, oh, you know what? These sorts of chemicals I see all the time in this topic. You know, this sort of polymerization I see all the time with these class of biological materials. This must be something. And that way you can kind of like narrow down your search and you kind of start seeing the similarities. And then you can start thinking about perhaps your own protocols and things like that. So, generally speaking. Okay, neat. Well, if nobody else has any questions or thoughts, I think we might wrap it up for the day.
you know what your professors feel like when everybody's just sitting in the room staring at them.
Okay, well, that was
So many thoughts, but yeah, just need time to process them. So is it okay?
Questions.
No, is it okay though if I just message you later with other things that I've thought of once I can sort of put together everything that you've said with the other stuff that I had? Is that all right?
yeah, definitely, and that goes for anybody. I mean,
Awesome.
and anybody who watches the stream in the future, if something was unclear, or if you have questions, or if you're like, but Jordan, this thing, yeah, I mean, like, obviously, feel free to reach out.
Thank you.
Yeah, of course. Yay! Okay, now we're more confused about more topics. Everybody, pat on the back. We're doing great. Learning the science.
Yeah, thank you, Beth.
Of course, anytime.
Thanks again.
Thank you
Thank
for
you.
listening.